Today I will talk about UFMylation and its role in resolving ribosome stalls at the endoplasmic reticulum. So UFMylation is a post-translational modification in which a small ubiquitin-like molecule called the UFM1 is conjugated to the target substrate via a cascade of 3 enzymatic reactions mediated by E1, E2, and E3 enzymes. Unlike ubiquitination, where there are hundreds of E3 ligases, in the UFMylation system, there is only one E3 ligase, which is this obligatory trimer composed by DDRGK1, UFL1, and CDK5, which is tethered to the ER membrane. And here they are, the cytosolic surface of the ER membrane is where modify its main target, which is a ribosomal protein called the RPL26. And this modification occurs exclusively on on the ribosome that are bound to the ribosome. And this is because both the conjugation machinery and the deconjugation machinery are tethered to the ER membrane. So the location of RPL26 is quite interesting. As you can see from this structural model of a ribosome docked on top of OST and Sec61 translocon, RPL26 is within this ribosome translocon junction. And here in blue, we mark the sites of UFMylation. So there are 2 lysines that are modified, and they are located at this C-terminus of RPL26, which is an alpha helix, here shown in red. And these 2 sites of modification are located in this position, and given where they are, it suggests to us that this modification might have a role in modulating the interaction between the ribosome and the translocon. So in order to understand what states the ribosome that are modified by UFM1 are found, we performed sucrose gradient fractionation, and we found that UFMylated RPL26 co-sediment with the EIF6 and NIMF, which are markers for 60S, suggesting that UFM1 is exclusively conjugated to free 60S. So it's an assembled 60S. So what's the reason why UFM1 is conjugated to free 60S, and what is its role if these ribosomes are not translating? So there is one situation in which cells produce 60S, and this happens when a ribosome stalls. Once a ribosome stalls, then you are left with this incomplete nascent polypeptide, which must be destroyed by the cell, and is targeted for degradation via the proteasome. The way it works is that the ribosome stall is split. Now you are left with a 60S with this arrest peptide attached to the tRNA. So this intermediate is recognized by ribosome-associated quality control mechanism, which trigger a stepwise process that recruits NIMF or IQC2 in yeast, which bind to the tRNA, and then recruits the C3 ubiquitin ligase called the listerin or LPN1, which ubiquitinates the arrest peptide and promote its extraction and subsequent degradation via the proteasome. Although this pathway has been extensively studied by many groups in the context of ribosome destall in the cytoplasm, very little is known on how ribosome destall during co-translational translocation are resolved. And this is complicated by the fact that while these arrest peptides are transversing the Sec61 translocon, this junction between the ribosome and the translocon is tight, and this junction shields this arrest peptide from the cytosol, where all the UPS machinery and the ribosome quality control components are found. Therefore, we ask whether the same ribosome quality control components that play a role in the cytosol are also involved in degrading this arrest peptide. And the second question was, are there other ER-specific components, such as UFMylation, which actually might play a role in modulating this junction and facilitating this location on the cytosol and the access of the UPS to the arrest peptide? So we developed stalling reporters in 2 different flavors, one that are targeted to the ER, and the other is a control that is cytosolic. And then the first thing we did was to treat cells with the proteasome inhibitors like bortezomib or lysosome inhibitor, bafilomycin. And we found that in the presence of bortezomib, we see accumulation of the arrest peptide that are targeted to the ER similar to what happened to the one that are targeted to the cytosol, suggesting that the ER stalled arrest peptide are degraded via the proteasome and not the lysosome. Then we test whether the same cytosolic RQC components that play a role in the cytosolic RQC are also required for this degradation of arrest peptides. And we did the knockout of DIMF and the Listerin one, which is the ligase that conjugates ubiquitin on arrest peptides. And we found that in both cases, you can see here, loss of NIMF or Listerin one stabilize this ER target arrest peptide to a similar extent to what is observed for the cytosolic counterparts, thus suggesting that the RQC is required for the degradation of this ER arrest peptide. However, one major difference between the cytosolic and the ER stalled peptides was the requirement for UFMylation. And this, you can see here, when we knock out UFM1 or other genes of the UFMylation pathway, we see that whereas there was no effect on the stability or the levels of the cytosolic stalled peptides, we found that the ER target peptides were accumulated, such that UFMylation is required for ER RQC, but not for cytosolic RQC. So the hypothesis is that UFMylation adapts this cytosolic RQC to destroy ER stalled peptide, but how it does this? So we propose a model in which UFMylation weakens the interaction between the translocon and the ribosome. And since UFM1 is a small protein of 83 amino acid, we speculate that the way it does is through a reader or a UFM1 binding protein that interacts with UFM1 and dislodges the ribosome from the translocon. So can we identify the UFM1 reader? So we in collaboration with the Harper Lab, Harvard, we did pull down of UFMylated ribosome and the mass spectrometry analysis. And by surprise, we found that the main interactors were components of the E3 ligase. So you can see here UFL1, CDK4AP3, and DDRGK1, which are the components of this trimer of this E3 UFM1 ligase, are among the highest hits. And so we asked whether actually the ligase itself could be the reader of this modification. And this was also validated by this biochemical assay done with sucrose fractionation, where now you see again, when you have a UFMylated ribosome on the 60S, you also see co-sedimentation of these three components, suggesting that they are associated with the UFMylated ribosome. However, interestingly, when we remove UFM1, so we have cells without UFM1, now you still have a 60S, but you lose the modification on the 60S, but you also lose the components of the E3 ligase that co-sediment with the 60S, suggesting that the UFM1 is required for this interaction. And it sets an unprecedented where the E3 ligase seem not only acting as a coder, a catalyst where it conjugates UFM1, but also as an effector because it binds to the product of its reaction. So given this persistent association between the E3 UFM1 complex and UFMylated ribosome, we decided to solve cryo-EM structures of the E3, sorry, I'm spoiling it. The E3 bound engaged with the free 60S. And then we identified three major states. State one, which we believe being the earliest state, we only see one of the three components of the trimer, UFL1, being resolved. And you can see here that it's bound to the tRNA transferase center. You don't see UFM1 here on RPL26, but you see the Sex61 translocon. Then we have a state 2, which we believe is an intermediate state. Now you have you resolve also another part of UFL1 and the other components of the E3 ligase. And you see that it's wrapping around the ribosome, going from the peptidyl transferase center behind here until close to the exit tunnel. So now you have UFM1 that is conjugated to the RPL26. You still have the Sex61 translocon. And then we found a state 3, which is basically similar to state 2, where you have this C-clamp shape on the ribosome. You still see UFM1, but interestingly, you also see a resolution of this helix of DDRGK1, which is positioned towards the exit tunnel of the ribosome. And every time we see this state, there is no Sex61, suggesting that they are mutually exclusive. and suggesting that the role of this DDRGK1 helix seemed to break off the ribosome of the translocon. So then I decided to perform some mutational analysis on DDRGK1, and actually the idea was to chop off this region, this domain that has a negatively charged amino acid that interact electrostatically with the parts of the ribosome close to the exit tunnel. So when we removed this, we did the same assay to look at this arrest peptide degradation at the ER by using our Stalling reporter. And as you can see here, these were done in DDRGK1 knockout cells, which similar to what happened with the UFM1 knockout. You see an increase of the arrest peptide when it's compared to wild type. Then we introduce the wild type here, and we have rescued the levels. So now arrest peptides are degraded, and we believe the way it does is through this dislodging mechanism. However, when we make this delta exit mutant without this region here, now, so we still see UFMylation, and we see the E3 complex bound to the ribosome. Now we don't see arrest peptide being degraded, suggesting that this exit binding domain of the E3 ligase is required for ER RQC. This work together is proposing a model which is still speculative because we haven't seen, and we are working on looking at this under a context of stalls and resolve cryo-M structures. But I'm proposing a model where there is a stall of the ribosome, you have split, you are left with the 60S and the arrest peptide, RQC factor binds, you have also the ligase, and I'm showing the 3 states. State 1, when you see only the UFL1 bound at the peptidyl transferase center. State 2, where you now have the UFMylation of RPL26. And state 3, where you see this DDRGK1 helix that dislodge or bump off the ribosome from the translocon. So this system is then turned off, and we have data showing that the UFSP2 is the deufimylating enzyme that is also anchored at the ER membrane, which remove UFM1 and disassociate all this complex, therefore rescuing the 60S to go over other rounds of translation. This was my last slide. I want to acknowledge my mentor, Ron Copito, and then Sam Gambin, that was a fantastic graduate student collaborating with me on the first part of the RQC story. Paul Da Rosa, that lead all the biochemical part and the structural work done in collaboration with the Roland Beckman lab, mostly Ivan Penchev. The mass spec done by Wade Harper at Harvard, and also collaboration with Yogesh Kolatu at Dundee University. Thank you.